my group does. I don't have a kinase-centric group, but I've been just blown away by the vitality of this field, and so I've enjoyed this conference enormously. We work in a couple of areas that all seem to kind of converge on kinases. One is trying to understand something about how phosphorylation regulates protein activity at a detailed atomic level. And that's what is going to be the main topic of my talk. If I have a little bit of time, I'm also going to talk about just a short story, a new direction that we're kind of going in, trying to understand something about kinase flexibility. This is mostly related to computer-aided drug design, which is not really the focus of this conference, and so I'm not going to emphasize that as much, but I do think that there are some connections to Alice theory and whatnot. Okay, so we've been seeing a lot of pictures of energy funnels and whatnot, and this is a much cruder version, of course. We've been trying to study phosphorylation for quite some time now, about 5 or 6 years, and the basic motivating question we had at the outset was just, when you add a phosphate to a protein, how does that modulate the energy surface of the protein? And then how does that drive changes in conformation and dynamics, which ultimately are related to function? So, of course, a lot of the effects of phosphorylation in a cell are mediated through modular interactions, protein-protein interactions involving like SH2 domains, FHA domains. That's not really what I'll be discussing. I'll be discussing what happens to, in most cases, a well-folded protein domain when you perturb it in this way. We've, in my group, looked at probably 15 to 20 systems or something like that at this point, trying to understand how phosphorylation perturbs the system, ranging from peptides, small proteins, big proteins, complexes. And usually when I give this talk, I go through 2 or 3 of those stories and try to explain how each story correlates with experimental data and whatnot. I'm going to try something a little bit different today. And try to give some more general conclusions that I've drawn from these various studies that we've done. It's always a little bit dangerous, of course, to try to draw generalities when you're talking about biology. There's always exceptions to the rule, and sometimes those exceptions are very interesting. But nonetheless, I'm just going to give you a sense of how we think about this at this point. So obviously, when you add a phosphate to a protein, you're introducing a large electrostatic perturbation. The predominant charge of the phosphate at physiological pH is -2. We'll talk in a moment about, the pKa is actually around 6.5, so I think that there is an issue about pH dependence of some of these effects. And of course, introducing a -2 charge in the protein is now kind of a unique thing. And so the thing that preoccupied us when we first got into this area was to what extent can we think about phosphoserine or phosphothreone as being something like an aspartate or glutamate, or does it behave fundamentally differently? And how does the physics change when you go from a -1 charge to a -2 charge? And that's really a dramatic change because taking the simplest model of solvation, the Bohr model, for a single ion solution, it says that the solvation free energy goes into the square of the charge. So in principle, this could drive things to look rather different. So in fact, the cost of fully desolvating a phosphate group is some large number, and I don't think it's exactly experimentally measurable, but it's well over 100 kcal per mole. And so it wasn't immediately obvious to us, would salt bridges involving phosphorylated amino acids be stronger or weaker than those involving aspar glue because of that enormous desolvation. Through a series of different experiments ranging from model systems to full proteins and whatnot, what it turns out is that, in fact, the interactions formed by the phosphorylated side chains are very strong with arginine, lysine, positively charged histidine. And in particular, the interaction between arginine and phosphorylated amino acid is probably the strongest type of salt bridge that you can see in any sort of normal protein system. I don't want to overstate this too much, but as a rough rule of thumb, if you're comparing the same interaction formed by glutamate with something positively charged versus phosphoserine with something positively charged in the same protein context and whatnot, the salt bridge involving the phosphate group will be about a kcal per mole stronger. That's just a rule of thumb. Okay. That's not, it doesn't sound like a huge number, but of course the phosphate group will often form multiple of these hydrogen bonds. The bidentate hydrogen bond with arginine is particularly stable, and is probably much more stable than the sort of strength that people would measure for normal sorts of salt bridges. So, okay, that's using glutamate as kind of a reference point for how do we think about these amino acids. And of course, but what's really happening is you're changing a serine or a threonine or a tyrosine, a neutral amino acid, to this -2 charge. So it's a really dramatic electrostatic perturbation, and you can wind up forming these very strong hydrogen bonding networks that can drive large conformational changes. This is just, this picture here, by the way, is just CDK2, and this is looking at a question of why does glutamate not mimic phosphoserine in this case. This has been known for a long time, but sometimes you can get constitutive activation. You can substitute a glutamate to act like a phosphorylated amino acid, but sometimes it doesn't work. This is one of the cases where it doesn't, and all the electrostatic calculations and whatnot bear that out. Okay, so of course what this implies then is that the phosphate side of it is only 1/2 of a phosphorylation switch. And in most of the systems that we've looked at where large conformation, substantial conformational changes are driven by phosphorylation, there's the other half of the phosphorylation switch, which is kind of the acceptor side, which usually consists, of course, of a series of positively charged amino acids. And CDK2 is kind of a remarkably well-formed cluster in the absence of phosphorylation. I personally would have thought that 3 arginine residues wouldn't want to be terribly close to each other, but even in the unphosphorylated structure, that's the case, kind of a pre-organized acceptor site for the phosphate in essence. These residues, as you would expect, are going to be well conserved evolutionarily, and this is just a second example here in myosin. The alternative to having a well-formed, pre-existing acceptor site for the phosphate, of course, is that you can have the site form after phosphorylation, and I'll show a little bit more of that in a moment. Okay, so the consequences of this is that phosphorylation is generally, and I'm thinking about how does this change the energy surface, generally speaking, is going to reduce flexibility in most of the systems we've looked at least. Increase stability in a local sense and oftentimes in a global sense, radiating outward from the site of phosphorylation as well. This is an example we were looking at recently, a protein called cofilin. That's an important actin-severing protein that's regulated by phosphorylation. This is one of the easiest simulations we've ever done. It's phosphorylated on the N-terminus on serine 3, and there was no structure, well, there is no structure of the phosphorylated form. There's NMR structures of the unphosphorylated form. And this is the conformation in the unphosphorylated form of this unstructured N-terminus. And you put a phosphate on it, and just like a tractor beam, a little cluster of lysines over here just pulls that phosphate in. And this winds up explaining how phosphorylation regulates the activity of actin, because this is an important part of the actin binding surface. Oh, and so that's structuring an unstructured portion of the protein, but running molecular dynamics on this, you can see that the overall disorder radiating, to other portions of the protein as well. The whole thing winds up being a little bit more rigid than before. The other, yes?
Is that a general statement, or is it focused on the active? The fact that phosphorylation induces rigidity? Because I found it be consistent that phosphorylation increases disorder.
Yeah,
I think Johnson also pointed out in one of the review that you can have either order to disorder or disorder to order transition.
I certainly believe that you can. And the driving force, of course, for going to disorder is the enormous solvation free energy of phosphate. So that aspect of it will drive towards disorder because the phosphate wants to be fully solvated, right? It may be that because we're primarily looking at cases of well-folded proteins where we're looking at conformational changes that, simply by looking at those types of cases, we're, biased in that way. The other consequence, of course, is that if you want to study a phosphorylated protein, using molecular dynamics, kinetic trapping can become an acute problem, even more so than it normally is. The energetic barriers connecting different states can become very large, and just in terms of the process of going from the conformation of like an inactive conformation to an active one, there's no particular reason why that has to be a short time scale. The bigger problem, to my mind, is not so much that kinetic trapping can be a problem, but how do you know? And in CDK2, we tried early on to just do an experiment where we took the unphosphorylated structure of CDK2 and added a phosphate to it to see, if it would activate, and nothing happened over a long simulation. And it was purely due to kinetic trapping. When we subsequently kind of mapped out the energy landscape using a different method, it seemed pretty clear to us why this was the case. Each one of these points on here is a local energy minimum on the potential energy surface. This is done in a implicit solvent. And so what you can see is that there's kind of two sort of macro basins here, one of which is the active form, one of which is corresponds, roughly speaking, to the inactive form, where the activation loop is pointed out towards solvent and kind of occluding the substrate binding site. And in between, we sampled as thoroughly as we could, and we couldn't find any sort of low energy pathway connecting the two. So it's a highly frustrated transition in this case. I should point out that the energy landscape for the unphosphorylated loop looks rather similar. It's just that the energies of these two basins wind up being inverted, and the inactive form winds up being the lower energy. By contrast, we were interested in understanding how phosphorylation of the regulatory light chain in myosin controls its activity. And this is one of the bigger simulations we've done. Is full explicit solvent, 300,000 atoms. And we had no particular reason to believe that that this should work. And yet, in this system, in just like 2 nanoseconds, we saw 20 angstroms of movement, and we're able to propose what I think is a pretty solid model for how this works. I'll show a little bit more of that in a second. So, it has nothing to do with system size. It's purely what are the energetic barriers. So this issue of, in some cases, phosphorylation induces virtually no conformational change. One of the first structures solved, I think, was isocitrate dehydrogenase. And the phosphorylated form was essentially completely identical. In other cases, there's dramatic conformational rearrangements. So we were interested in some of these cases where there are these very large changes, and how does it work. And in retrospect, there's a lot of literature out there that makes this stuff kind of sound mysterious, but I think in retrospect, the picture is fairly simple. So this is another molecular motor, myosin. The two head groups, which we should be calling feet. and eventually going out to a coil-to-coil. So the regulation here occurs not on the heavy chain itself, but rather on a calmodulin-like light chain that wraps around a particular joint down here. And this is a rough structure of the unphosphorylated form solved by electron microscopy. And the regulatory light chain is phosphorylated on its N-terminus, and this has been known for a long time in smooth muscle myosin to kind of act like an on-off switch. And the essence of our model for how this works is, when you add the phosphate to the regulatory light chain, it's kind of like, when you add calcium to a calmodulin domain, you can use conformational changes as it wraps around the helix of the heavy chain. And this winds up torquing that joint, this flexible joint in the helix, and winds up relieving an auto-inhibition of these two heads by sort of pulling them apart from each other. And then, we haven't reached the fully activated form in a short simulation, but I think this is probably fairly close to the essence of what's going on here. Another example recently is ARP23. This is an important protein in actin regulation. And it just very recently it's turned out that ARP23 is regulated by phosphorylation. This wasn't known for a long time. Phosphorylation is necessary but not sufficient for it to do its job. And the phosphorylation site at first looked a little bit mysterious to me in that it's a lot more buried than I'm used to seeing, way kind of deep in here on ARP2. I don't know exactly how the kinase gets access to it or if it phosphorylates it before it forms. I have no idea. But in terms of what it's doing, how it's doing its job, I'll pull up a movie here. This is just a morph from the, and there is no structure of the phosphorylated form. So this is not like we're, doing biased MD from one state to another. We're, insofar as possible, trying to make predictions about systems where we don't know the answer already. And so this is starting at the unphosphorylated form and then showing, the essence of the conformational changes that occur over a few first few nanoseconds, again, a short simulation, about 10 nanoseconds so far. The phosphorylation site is right in here, and this is aligned in such a way so that we're keeping this subunit here roughly fixed. And so you can see the rather kind of dramatic changes in this subunit organization. And of course, then, in retrospect, it all seems blindingly obvious. The phosphorylation site is situated right here because it's sitting right next to this long helix, which acts as a semi-rigid element, and is again kind of torquing that thing and driving these long-range conformational changes. And this is all being mediated, of course, by specific positively charged amino acids in this vicinity. Okay, and the last thing I want to throw out, I don't have any reason to believe at this point that this has any biological relevance, but I still think it's worth pointing out. And that's that the phosphate group has this pKa for the first protonation is about 6.5. And so, in principle, depending on the environment, it could either have a -2 charge or a -1 charge. And what this implies, and that's about the same as histidine. So, when you think about pH effects in biological systems, you immediately think histidine. So as this thing accepts a proton in peptide systems, it's been shown very clearly that it changes the conformational ensemble. Okay. And so I forget who did the experiments on this, but we simulated this phospholipeptide at different pHs, and you can see that you can capture conformational differences that are also captured by the experiment by comparing to NMR observables. And this is just looking at the strengths of these hydrogen bonds as a function of protonation state and how it's modulated by pH. So I don't know if it's biologically relevant, but certainly in vitro, I would expect that even in protein systems, you might see some effects of this sort. And finally, behind all this, for the computational people who are working in these types of systems, we spend a lot of time worrying about the electrostatics of the phosphate group and whatnot. And when we initially tried to use things out of the box, various charge sets and O kill S, or this is a semi-empirical quantum mechanics, we got results that just were terrible. And so, although these changes in the partial charge, it's from about 1.4 to 1.9, using different charge sets that are out there, it's not just a minor difference, and they can lead to completely different qualitative conclusions. So just be careful, get these things out of the box. So I guess the last thing I wanted to say here is that we've had a couple of systems now where we've made predictions that have been tested experimentally, and we've published a couple of these. But we're very much interested in looking at other systems where we can work together with experimentalists. Because I think Susan's absolutely right that we need to test these sort of models that we're developing. Okay, the second and shorter story that I was going to talk about has to do with kinase dynamics, and I don't want to go into details about inhibiting kinases and whatnot. The story that I'm going to talk about is PI3 kinase. So not a protein kinase, and how the dynamics changes among isoforms, which I think might have more general interest. The second reason I want to show this is because of the methods that we're using. We heard yesterday about accelerated MD as a method for trying to increase sampling. And as I've already argued, this is a very serious challenge when you're dealing with phosphorylated systems, for example. And so we've been coming at this from a little bit of a different direction. Okay, so PI3 kinase. This is a picture from one of Kevon Shokat's papers. They, through fairly serendipitous means, discovered this inhibitor that showed good selectivity for PI3 kinase. And what this picture is showing here in blue are a whole bunch of crystal structures of PI3 kinase that have been solved previously. And if you just looked at these crystal structures, you would say that this inhibitor would not fit. Okay, this PIK39. And when they eventually solved the structure of this inhibitor bound to PI3 kinase, it turned out that there was a pocket that was induced and that they had previously not suspected was there. So one thing that computational simulations, molecular dynamics, and whatnot are good at, in my opinion, is identifying what parts of a binding site, for example, are flexible and what parts aren't. So what I'm showing you here is this is a Monte Carlo simulation. In this particular case, it's only on the side chains. I'll show you a more complicated one in a moment. And you can kind of see the essence of why this is a more efficient way of sampling than molecular dynamics, at least in principle. And that's because we're making all of our moves in torsion angle space. So you can move from one sidechain rotor to the next in just a single move. And so you get very effective sampling in a short period of time. And what we were immediately able to see was that this methionine that formed this induced pocket was in fact extremely flexible, and we could identify in this ensemble conformations that were productive for binding this particular ligand. So that's kind of a conformational selection type paradigm. Okay. So the more interesting aspect of this, though, is that there are 4 different isoforms of PI3 kinase, alpha, beta, delta, and gamma. And all of them have exactly the same amino acids in the binding site contacting this ligand and other ATP competitive ligands. But K-Von's people were able to develop compounds that had selectivity of up to a factor of 1,000 for one isoform versus another. And this is something you see in many cases, that you can get selectivity even when the first shell of residues is entirely conserved. And so we were able to explain this through dynamical effects, second shell amino acids changing the dynamics in the first shell, which in turn is related to the SAR. I'm going to show you the pair that is the easiest to understand, and it's kind of like blindingly obvious why there's a difference. This is the same pocket here where this is kind of the selectivity pocket sandwiched between the tryptophan and the methionine. And this is gamma and this is delta. In gamma, there is in the second shell this glutamic acid that winds up forming a hydrogen bond to the tryptophan, and you can kind of see in the simulation here that it's largely holding the tryptophan in place, and in part kind of keeping this pocket kind of closed. When you go to delta, when you go to delta, it turns out that that particular amino acid is mutated in methionine. Now the tryptophan is sandwiched between 2 hydrophobics, and you get a much more dynamic pocket, which opens up much more frequently, meaning that the free energy of opening that pocket, in essence, is reduced. Okay. So this is, I think this issue about how do you gain selectivity, between very closely related kinases is very interesting. This, by the way, is the equivalent of the glycine-rich loop. Although it doesn't have any glycines in PI3 kinase. And we've been looking at related, for example, SARC family kinases to again see some dramatic differences in the dynamics in this region, in the glycine-rich loop region of the kinase. And I'll stop there and just thank the students and postdocs who did this work and collaborators and funding the course. Questions for Ben? So we have a variety, we get many different types of computations to assess this. These included umbrella sampling and explicit solvent, quantum mechanics with SCRF, treatment of solvent, calculations on model post-gas binding systems, etc. As well as these pairs where people have engineered in glutamates and should try to mimic phosphorylation and things like that.
About the phosphorylation, what is the change in volume due to desolvation? And has anybody measured the change in convertibility?
Sorry, the change in what?
In volume.
In volume? Well, it is somewhat bulkier sterically, not vastly, but obviously the phosphate is breaking down an extra oxygen.
Yeah, but when you consider all the hydration, is it positive change or negative change?
Positive.
Positive.
Yeah.
Has anybody measured the compressibility?
Compressibility? I don't know. I don't think so. I'd be interested in the results, but I don't know.
So I thought that was a really interesting observation about the CKA issue, and you have the occasion to put a phosphonate into proteins instead of a typical phosphate, and you can either put a CH2 phosphonate, which will raise the pKa another unit or so, or you could put a CF2, and you do see some functional differences there, and that's thought, at least we've attributed some of that to the pKa difference. Would it be difficult for you to model in phosphonates?
Not the slightest.
Because then one could sort of computationally assess how important that is.
Yeah, I think that's a terrific sensitive way to test this hypothesis. I mean, biologically, I just don't know how to go in and ask what proteins, if any, might be, might this be relevant to? I don't know. I mean, obviously, if you had phosphorylated proteins going into, an endosome or something like that, then, there very well could be pH-dependent changes.
So, related to the previous question, so have you thought about other forms of modifications like sulfonation of cysteines and?
Oh yeah, acetylation, methylation, etc. Yeah, absolutely. There's very little known right now, which is great from a computational perspective, but also slightly frightening, right? You don't want to do something completely nonsensical. I mean, the other one would be nitrosylation. I mean, it's not a change in charge, but it's very interesting for other reasons.
But in terms of parameters, you have to do a lot more work there if you want to.
It's a little bit of work, but it's not that big a deal.